Bismillahirrahmanirrahim. In today's lecture, we will study the gross and ultrastructure of cell wall. On the basis of development and structure of plant tissue, three layers in the cell wall can be distinguished: the middle lamella, the primary wall, and the secondary wall. The middle lamella is found between the primary cell walls of the neighboring cells. It cements these walls together in a tissue in supporting tissue, which have thick walls. It may also fill the intercellular spaces. It consists of pectic substances. It can be dissolved by enzymes, pectinases, and this procedure is called as maceration. The primary wall is the first true cell wall which develops on the new cell. It develops in cells or portions of cells which are still growing and is composed of mostly celluloses. The secondary wall is formed on the inner surface of primary wall. It develops in cells which have ceased to grow. It is anisotropic and layering can be observed in it. In tracheids and fibers which have very thick walls, its three layers can be observed. These are the outer S1 layer, the middle S2 layer, and the inner S3 layer. Of these layers, the middle S2 layer is the thickest. In some cells, there are more than 3 layers. In that case, some authors use the term tertiary wall for the innermost layer of the secondary wall. Ultra or fine structure of the cell wall. This structure has been extensively studied because of its importance in fiber, paper, and other industries. The cellulose molecules consist of long chains of linked glucose molecules. These chains are arranged in bundles called as micelle, singular micelle. The number of glucose residues in cellulose molecule is 500 to 10,000, and the length of these molecules varies from 0.25 to 5 micrometer. In each micelle, cellulose molecules lie parallel to each other. These micelle are packed to form microfibrils. Microfibrils in turn are packed with little or no spaces to form bundles called as macrofibrils. The spaces between the microfibrils are called as capillary spaces and are filled with water and other chemical substances like lignin, cutin, and hemicelluloses, etc. In cells of supporting tissues, the interfibrillar space is hardened by the deposition of solid substances. This process is called encrustation. The encrustation of lignin is called as nignification. Some other substances such as suberin, cutin, waxes, quinones, tannins may also encrust the cell walls. This diagram depicts the ultrastructure of the cell wall. These are two glucose molecules linked by beta 1,4 glycosidic linkage. This is a portion of the cellulose molecules containing glucose chains. These chains form elementary fibrils. These elementary fibrils are aggregated to form micelle, and micelle are aggregated to form microfibrils. These microfibrils are aggregated to form larger unit, which is called as macrofibril. The orientation of microfibril is more regular in secondary walls than in primary walls. In primary walls, their orientation is essentially at right angle to the long axis of the cell. As the cell grows, orientation of microfibrils becomes longitudinal, that is along the axis of the cell growth. Thus, the orientation of microfibrils determines the direction of the cell growth. When fibers are irregularly arranged, the cell enlarges in all directions. Dear student, this concludes our lecture about the structure of cell wall. Thank you.